All right. Hi, everyone. Welcome to this new edition of the Worldwide Neuro Dev Forum. Good to know you guys are out there. And as you see, we have a special configuration today with three speakers. So before I introduce our main speaker, Randy Platt, we have this special session that I hope will become more common. I'd like to encourage every junior person out there and every person that you know to participate in these so-called flash talk. The idea is that typically a PhD or a postdoc would present his or her work in 3 to 5 minutes and get some level of exposure before the main talk. So today's inaugural speaker is Chris Fell. Chris studied for his master's at the University of Leicester in the UK for his thesis, investigating the genetics underlying Drosophila circadian rhythms in the lab of Ezio Rosato. He then moved to Vienna for his PhD, joining the lab of Vanja Nagy at the Ludwig Boltzmann Institute for Rare and Underdiagnosed Diseases, where he works on the genetics of rare intellectual disabilities. So Chris is going to tell us in a flash format about his PhD work. Go ahead, Chris. Stage is yours.
Yeah, thank you very much. Let me share my screen. So I hope you can see this properly.
Yeah, it's perfect.
Okay, great. Yeah, so good afternoon, everyone. Thanks for the introduction. Yeah, my name's Chris Fell. I'm a PhD student in the Ludwig Boltzmann Institute for Rare and Undiagnosed Diseases here in Vienna, in the lab of Vanja Nagy. I'm going to tell you today about my primary PhD project centering around the gene FIBCD1. So this is a schematic showing FIBCD1. It's a type 2 transmembrane lectin protein, meaning that it binds sugars, and it's predicted to have an endocytic function. There's not much known about FIBCD1 so far. There's been some work showing that it has a role in innate immune response to fungal infection, but there's nothing known about FIBCD1 in the brain or anything to do with neuroscience. So what turned our attention onto FIBCD1 was basically its expression pattern. This is qPCR data from human visceral tissues, and we found that the brain is the third highest expressing tissue in the body. And within the brain, the hippocampus is the highest expressing region, leading to the question of this project, basically, what is a fungus binding receptor being expressed so highly in the brain? To cut a long story short, we've basically been able to show that it recognizes a component of the brain's extracellular matrix, which is depicted here. So the extracellular matrix of the brain is partially composed of these conjoined sulfate proteoglycans, or CSPGs, which are themselves consist of a core protein and a branching glycosaminoglycan, or GAG chain. And these consisting of conjoined sulfate in this case. And these conjoined sulfates can be differentially sulfated, and these three shown here are very tightly regulated in space and time during development. And what we've basically shown is that FIBCD1 binds and recognizes conjoined sulfate 4S specifically. So when we do Western blots of lysates from the hippocampus of wild type and knockout mice, for these three different conjoined sulfates, we find an increase in signal of the conjoined sulfate 4S here in the knockout hippocampus. This has consequences on the behavior of the mouse. So this behavior test I'm showing you here is known as inhibitory avoidance. How this works is that you have a box consisting of a light and a dark chamber, and you put the mouse in the light chamber and time how long they run into the darkness, which they prefer. Within the dark chamber, they receive an electrical shock, and then a mouse with a functioning hippocampus then associates that dark chamber with the painful stimulus. So the training period is the first time that you put the mouse in, and then the test period is the same, is the mouse put back into the light chamber after 24 hours. And we find that the wild type and the heterozygous mice are much more reluctant to run back into this dark chamber, whereas the knockouts basically run back in much faster, indicating they have not associated this dark chamber with the painful stimulus properly, and they do not have a properly functioning hippocampus in adult life. Our collaborators profiled the hippocampus electrophysiology logically, including long-term potentiation shown here. This is hippocampal slices profiling the evoked response of the Schaefer collateral pathway. The four conditions we have are the wild type and the knockout slices pre-treated with a negative control enzyme called penicillinase, which has no endogenous substrate in the brain, or pre-treated with this enzyme called chondroitinase ABC, and this targets these structures for degradation. The idea being that we break up this accumulation that we already showed in the hippocampus. So this is measuring evoked response to stimulus at time 0, theta burst stimulation. We get this synaptic remodeling and long-term potentiation, so an increase in the evoked response. This dark blue line, which is the wild-type penicillinase, which is the wild-type baseline, potentiates nicely over time. The knockout, which is the dark red, has an impaired long-term potentiation, which actually reflects the behavior phenotype we see here because the synapses have not remodeled properly. The wild-type hippocampi pre-digested with this chondroitinase ABC shows impaired long-term potentiation is the blue line down here, which has actually been previously described. But then what we were very pleased and slightly surprised to see is that the knockouts pre-treated with this chondroitinase actually have a full rescue of this long-term potentiation phenotype. So this brings me to the end. Basically, our working model for what's happening is it's known that during activity-dependent synaptic remodeling, you get digestion of these CSPGs, for example, by matrix metalloproteases shown here. What we think FIB-CD1 is doing is binding and endocytosing these CSPGs, although this endocytosis and degradation we haven't actually shown yet. And what we think is happening is that in the knockout, this endocytosis doesn't happen. You have this accumulation of CSPGs, which inhibits synaptic remodeling. So thank you all for your attention. Thanks to our collaborators. This was a highly collaborative project between our lab and the Paniga lab. And especially thanks to Anna and Francisco for the EFIS data. Yeah, thank you all for your attention, and I'm ready for your questions.
Yeah, thanks, Chris, for driving us through this. We have time for one or two questions. I'll ask one, and maybe Randy has one too. What about the locus of the sulfatation? You focused on the 4S. Anything known about the 0S, the 6S, and why would that one be more interesting than the other ones?
Yes, so it's a good question. It's known that these 4, 6, and 0S are tightly regulated during development. So the 6S is expressed very highly very early during development. And then in the so-called critical period of the hippocampus, which is when it matures, you get a switch of chondroitin sulfate 6S being high to chondroitin sulfate 4S being high.
Okay.
So what exactly they're doing is not really known, but it's clear that 4S is key to sort of the maturity of the brain and the hippocampus.
Cool, thank you.
Chris, really interesting work. I do have a question. I'm interested in the connection between the CS4S endocytosis and how it leads to synaptic remodeling. Do you know much about the mechanism of that process?
So it's not so much the endocytosis that leads to the remodeling. We think it's more the clearance of this like accumulated CSPGs here. So it's known in the literature that dysregulation of these CSPG levels is important to synaptic remodeling. So yeah, it's more the accumulation we think is the problem, and then this is almost like a garbage collection by FIB-CD1 into the cell. Yeah, but I'm just speculating. We don't know that for sure.
All right, Chris,
thank you very much.
Thanks a lot for sharing this. Thanks a lot for being our first pre-speaker. Good luck with the project, and hope to see you around.
Great. Thanks a lot for having me.
Thanks, Chris.
All right. Wow, the transition actually worked. All right. Randy, the time has come now for you to walk on stage. So welcome, first of all. A few words about you before I leave you on your own. Randy studied biomedical engineering and chemistry at the University of Utah. In 2011, he obtained his MPhil from Imperial College London in material science, and then a PhD in 2016 from MIT in biological engineering. He did a joint postdoc at Harvard, MIT, and abroad, and joined the ETH Zurich, but in Basel in 2016 at the Department of Chemistry. Randy's work is characterized by a beautiful combination of state-of-the-art molecular tools with state-of-the-art biological questions. To a large extent, his work has been focusing on non-neuronal models. But as you will see, and as I think he will tell us about, as I hope you will understand also, these tools are quite versatile and flexible, and I think will be of interest, or are already of interest, to many of us in the neuro or in the developmental field. Randy, thanks a lot for being here. It's really a pleasure to have you. We're aware of the fact that you're not neurocentered, as I mentioned, but I think it's a strength for today's purpose, and really humbled to have you here.
Thank you, Denis, for the really nice introduction. I'm humbled to be here. I'm humbled by the invitation, and very excited that all 88 or hopefully more of you are out there tuning in. I hope you're all safe and healthy where you are. Again, my name is Randy Platt. I'm an assistant professor at the ETH Zurich in Basel. The department is Biosystems Science and Engineering. And I'm going to spend the next almost hour telling you about some of the work we've been doing over the past 4 years or so about developing the CRISPR toolbox in order to engineer biology. And as was said, the talk is going to cover some neuro topics, some non-neuro topics. My lab heavily works in this area, but none of it is quite published yet. So in the next few years, you'll hopefully see more and more. And so since we do a lot of deep technology development, the way I think I can convey this knowledge to you in the most sensible way is going to be more of a highlight reel of some of the projects that we're working on and how they can be used to engineer mammalian cells and the mouse brain in particular. So let's just get right into it. One of the greatest scientific achievements of our time was sequencing of the human genome. It not only brought into view the over 3.5 billion DNA bases that comprise the blueprint of life, but it sparked a revolution in the biomedical sciences, right? We better understand genes, introns, exons, non-coding, we know enhancers, we learned a lot. And what that did is it also sparked an additional revolution in the biomedical sciences, which is DNA sequencing, whereby we not only have one human genome now, we have hundreds or thousands, and we can start to compare and contrast individuals and identify genetic alterations that are linked with different human phenotypes. Some of them which can result in disease. And this is what I'm showing you here, where over time on the x-axis, we've found more genes that we can associate with different human genetic disorder. There's been this rapid increase, and then in 2017, that number exceeded 6,000. So 6,000 of the over 20,000 protein coding genes in the human genome are associated, not quite causal yet, but associated with a genetic disorder. And if you think about how research is done today, it's usually one lab or even communities that study one or a small number of genes for an entire career. Think of P53. This is a gene that's been the subject of decades of research in dozens of labs. So how can we get close to studying this number 6,000? How can we identify which ones are causal, understand the molecular mechanism, and develop genomic therapies to treat them? And this is the core motivation of my lab, where we use genome editing tools to try to meet this pace of discovery. Can we go from association to causal, mechanism, and genomic therapy much faster? And we approach this from a technology development standpoint. And in this day and age, of course, we use CRISPR to do that. Most of you at this point have probably heard about CRISPR. And just to reconcile any mysteries, CRISPR isn't just CRISPR-Cas9, it's a whole prokaryotic adaptive immune system. And since my lab uses many CRISPR proteins for biotechnology, I want to quickly remind you this process and how CRISPR is a treasure trove of proteins for engineering biology. So the CRISPR-Cas adaptive immune system starts with an infection. The infection of a bacterium with a virus. So here is a bacteriophage injecting its, in this case, double-stranded DNA inside of a cell. And then the first step of CRISPR adaptive immunity is recording. This is a process whereby CRISPR-associated proteins physically excise a piece of DNA or RNA from the invader and insert it with their own genome, where it serves as a molecular memory of that infection. And within this location in the bacterial genome, it's called a CRISPR array, and these colored things are called spacers, and each one of them are derived from a different invader. So it serves as this molecular memory or this fossil record of previous infections. At a later point in time, these memories are recalled as one long pre-mRNA transcript, which is subsequently cleaved into individual RNA molecules that are guided by CRISPR-associated enzymes like Cas9 back to the target, where they make DNA double-strand breaks or perform other modifications that defend that that provide immunity for that bacteria against that virus. And so these three steps, record, process, and perturb, are executed by different enzymes that allow us to manipulate cells, and that will be the topic of the remainder of this talk. And I'm going to give you 3 research vignettes in areas that we work in. The first one is in vivo genome editing and functional genomics, engineering genes and genomes inside living animals. The second is going to be transcriptional programming, that is using CRISPR tools to not target one gene, but dozens of genes towards massively programming cellular transcriptional states. And in the third part, how we leverage the CRISPR recorder in order to turn cells into transcriptional recorders, that is, living cells that we hope to use as diagnostics in the future. But first, let's start with in vivo genome editing. So major advancements in the biomedical sciences have been made for engineering genes and genomes to study gene function in living animals. The first big innovation was gene targeting. This is the process by which you engineer embryonic stem cells and plant them into blastocysts, and through implantation into donors and breedings, you can engineer these transgenic mice. It works about 35% of the time, and they take years to make. It won the Nobel Prize. It's incredible, incredibly powerful technology. A huge innovation was made through the process of figuring out that one can inject CRISPR-Cas9 reagents directly into single cell embryos, where the mutation can be made in the genome, and then that single mutant cell gives rise to a whole mutant animal. Many of you, probably all of you in the audience, know this, and this is routinely done in all transgenic facilities throughout the world. But the main innovation here was that it reduced the amount of time that it took to engineer this animal from years to months. And that's getting us closer to understanding these 6,000 genes that are associated in the disease. However, I would argue that the fundamental bottleneck is still the fact that you have this transgenic animal model at the end of the day. It's okay to make one or dozens of these, but it's impractical to really have 6,000 or 6,000 or even more if you look at unique variants. And so in the past, in order to sidestep this fundamental bottleneck, we came up with the idea of, rather than engineering mouse colonies, what if we just deliver CRISPR-Cas9 directly in vivo to the cells and tissues that we care about, where the mutation would occur, and then we can understand the phenotype of that genotype very quickly, where in principle that would reduce the amount of time for engineering or creating these mouse models from months to weeks. And this data is quite old now, but I think it's a nice introduction to the functional genomics part. So let me give you a quick example of how this works. We engineered the special mouse line, which is a Cre-dependent Cas9, and we can deliver the guide RNA and Cre via a viral vector like AAV. So here's an example of a stereotactic injection of this AAV into one side of the mouse brain. This side was injected, this side wasn't. The Cre expresses, which in turn turns on Cas9 on this side of the brain. And we targeted NuN in this experiment. So the guide RNA targeted the RBFOX3 gene, which should knock out NuN. And here's a merge of 2 channels. And the take-home message here is that more or less every cell that gets infected with the virus no longer expresses the target protein. And this was a very exciting experiment. And what this basically told us is that rather than making a transgenic animal, what we can do is we can pick a gene, design a guide RNA, make a virus, and get a complete spatial temporal knockout in a matter of weeks in the lab. And that's getting us much closer to studying these 6,000 genes. That was a nice proof of concept, but my collaborator, Sidi Chen, and I wanted to take this one step further to see if we could use in vivo genome editing to model multigenic disease processes. And we set out to model or recreate lung adenocarcinoma, which is the most common cancer type in the world, results in about 1 million deaths each year. And because of this, we know the human genetics quite well. And so in order to recreate lung adenocarcinoma, what we wanted to do is create a triple mutant. That is, we wanted to knock out 2 tumor suppressors, P53LKB1, as well as knock in a gain-of-function mutation in KRAS. In order to do this, we had 1 guide RNA each for these knockout mutations, and then for KRAS, we have both a guide RNA and a donor template to swap out the wild-type allele for the gain-of-function allele. And so if you inject these in the lung, you can get robust expression. And then if you wait about 2 weeks to 2 months, take out the lung, stain them, you start to observe all these lung adenocarcinomas. I wasn't a cancer biologist at the time, so what I did is we took these slides to a clinical pathologist, and he said, indeed, these are lung adenocarcinomas, probably driven by KRAS, ranging in severity from grade 1 to grade 4. We stain them for the appropriate clinical markers, and these animals fully recapitulate what would be expected of a mouse model of the human disease. And why we thought this was very exciting is we could go through this process from gene selection to viral vector to in vivo phenotype in 1 to 2 months. And if you think about the classical method of making a triple transgenic, it's not that you make the triple transgenic, you start with each individual mutation, you intercross them, and then eventually get your triple transgenic. So we can basically reduce the amount of time for studying these models by about 5 to 10 years. In this very old paper, we not only did that, we showed that we could deliver the guide RNA using various delivery modalities, from nanoparticles to AAVs to lentivirus to all the different tissues. And we found this to be a very exciting model. So when I started my lab in Basel a few years ago, I wanted to extend this concept and increase the scalability a little bit more. It was interesting that we could target 1 or 3 genes, but we weren't really getting that much closer to 6,000. And the only way to do this in a scalable fashion is to apply the concepts of forward genetics. And this is the process by which, where you ask many questions at once, you need really intricate cellular selection systems, which I'll talk about in detail. But the point being, rather than mutating 1 gene at a time, why don't we mutate a whole bunch of genes, potentially even genome-wide, and ask the question, which gene is important for a phenotype that I care about? And so here's our first effort to try to answer that question. This was in collaboration with Sidi Chen and his postdoc Ryan Chow at Yale and my PhD student Florian Schmidt. And the question we were asking was regarding the induction of glioblastoma. So the originating cell type of glioblastoma, which is a primary brain tumor, is an astrocyte. So the question we're asking is, which gene in an astrocyte, when mutated, will allow that cell to transform and proliferate into a glioblastoma? We didn't start off asking this question genome-wide. We used human pan-cancer data to identify a putative set of tumor suppressors. So we mined this pan-cancer data set and identified the 50 most commonly mutated tumor suppressors across human cancers. For each of these putative tumor suppressors, we designed 5 guide RNAs, which you can then synthesize on a chip. You can clone them into a plasmid library where each plasmid harbors a unique guide RNA. For the experts in the audience, this is what the plasmid looks like, where we have this GFAP promoter, which is an astrocyte-specific promoter, which should drive CRE and thus turn on Cas9. And then we have this P53 guide RNA to sensitize the cells. And then we have one of our library guide RNAs to ask the question, which one is important? We can then use this plasmid library to make a virus library. And then we can inject the virus library into a living mouse brain very carefully, such that mostly single cells get single infections and thus single perturbations. So this cell got a red perturbation, this one got a blue one, and this one got a yellow one. And then over time, the cells that obtained a mutation that is important for proliferation will start to overtake the cell population. And here's an MRI image showing that indeed that these viral vectors lead to induction of glioblastoma. The mice die with typical Kaplan-Meier kinetics. Then if you take out these tumors and sequence all the mutation and ask the question, which ones are found more frequently, meaning that they're important, this is what it looks like. I'm showing you this as a summary of the data. It's about a 9-figure paper, so I won't go through it in detail. I just want you to appreciate the abundance of information we have, where On the x-axis, we have different tumors, and on the y-axis, we have different genes, the putative tumor suppressors. If that box is colored, that gene was mutated in that sample. What you can see is that some genes are very important, while others not so much, and they're colored by their functional category. And so with this, we get a wealth of information. We can understand the tumor suppressor landscape in glioblastoma. And just to quickly convince you that this mimics the human patient situation, here's a scatter plot where on the x-axis you have our data, the frequency of mutations in these genes, and then on the y-axis, the frequency of mutations found in human patients. And what you can see is that at the low end of the spectrum, not much is going on, but at the high end, we can pick up some of the major players known to drive glioblastoma in human patients, like P10, NF1, RB1, and so on. So we can really identify these main drivers. To the astute members of the audience, clearly you'll see this gene here and this gene here. B2M is found high in our sample, not in human patients. But it turns out this B2M gene is very important. It's part of this MHC class II complex, which basically allows these tumor cells to evade the immune system. So it is important for tumor genesis, but not so specific to glioblastoma. CDKN2A, on the other hand, was a false negative. It's a gene we missed, and the reason it turns out is CDKN2A has a very complex gene expression regulation. It has a bunch of cryptic promoters. So basically our guide RNAs were not knocking it out appropriately. What else we can do with this data set is start asking questions about co-mutational pairs. How often are the same genes found mutated in the same samples? This is called co-occurrence, where you see this on the X and a statistical metric on the Y. And what you can see is that we can identify these co-mutational pairs. And what this does is, in the top set of this list, it identifies co-mutational pairs that defy patient subtypes of glioblastoma. So there's a lot of value there. And also it finds synergistic pairs, which open up strategies or entry points for cancer therapies. So this was a very exciting study, which we followed up to show that it is generalizable. It's not just specific for glioblastoma. We can also do it for hepatocellular carcinoma just by changing the promoter that drives CRE and delivering it a slightly different way. So here's the liver with tumors, and again, the co-mutational landscape. So that was great. This was our first attempt at performing in vivo functional genomic screens. These screens require a specific cell selection, where what I just showed you is proliferation. A cell goes from one cell to lots of cells, and you use deep sequencing in order to count this. But there are other cellular phenotypes that you can use screening to interrogate, such as senescence and cell death, fate specification, or other marker-based readouts, as well as things like migration, where cells move. And so for the next few slides, I'm going to tell you about how we tried to use CRISPR screening in vivo to ask questions about fate specification. So this work was done in collaboration with Paola Arlata and her fantastic postdoc, Ashwin Shetty, who is hopefully listening in today. And we asked this question about colossal projection neurons in the upper cortex of the mouse brain. And so for those of you that don't know much about this process, I'll briefly give you neocortical development 101, where the embryonic mouse brain looks like this. You have the ventricle zone here. And then what we do is we basically try to inject virus here to infect this ventricular zone, or these radioglial cells, which are these progenitor cells that asymmetrically divide at different points in time to give rise to different cell types with different identities, which radially outward migrate and form these very complex but radially organized cell structures that in each layer has a unique identity of cell types, which project to a unique region and have discrete functions. While we know the fate specification, or the master regulators that orchestrate differentiation of some of these cell types, we know very little about others, such as these red cells here, which are called colossal projection neurons. These are these pyramidal cells that asymmetrically project to the contralateral side of the brain, and they're important for bilateral integration of information. So the question we're asking is, what are the master regulators or transcription factors that orchestrate this process of radial glial cell becoming a colossal projection neuron? And in order to ask this question, which is quite complex, everything happens very quickly, we needed to come up with a scheme that allows us to both knock out genes. So what's happening is that at E12.5, you're only starting to get this ventricle zone structure. And so what we found is best is if we electroporate our plasmid library at E12.5, we can infect these radioglial cells, which leaves about 24 to 48 hours for the gene to be perturbed, the protein to be depleted. And then when the radioglial cell makes the fate choice to asymmetrically divide to become a colossal projection neuron or another cell type, that perturbation exists. And then in pups, we can isolate these cells and determine which mutation was important for that process. But just to validate that we could achieve genome editing in this very dynamic time frame, we performed a pilot experiment where we created this plasmid that expressed both TD tomato and a guide RNA. We electroporated it at E12.5, and then 3 days later, we performed FACS on both TD tomato positive. We just performed FACS on TD tomato positive cells, and then performed deep sequencing on the target to see if we could mutate within that time frame. So here's an immunofluorescent image showing that indeed we can electroporate this layer within the cortex. Here's a FACS scatter plot showing that we can isolate these cells. And here's the deep sequencing result for guide RNAs targeting 2 different genes. One is SAPB2 and another one is CUX1. And in these 3 days, we can achieve editing rates of about 80%, which is surprisingly good. So this gave us enough confidence to proceed with the screen. Here we designed 384 guide RNAs targeted to about 50 different genes that were selected based on existing RNA-seq datasets. We subcloned this library into a plasmid library, which we electroporated at E12.5, and then isolated TD tomato positive colossal projection neurons at P0. And then we analyzed through deep sequencing the genes or guide RNAs that were over or underrepresented in the final endpoint. So imagine that if you targeted a gene that was essential to the cell, it died, it would look like a depleted gene. An alternative is that gene was important for the differentiation of colossal projection neurons. And here is the screen result of this pilot library, where on the x-axis you have the genes alphabetically sorted, and on the y-axis the statistical metric of whether or how enriched that guide RNA was in that end population. And all hits were depleted, so they seemed to be important for colossal projection neuron development. And this was a pilot result, but let me just try to convince you that the genes that popped up have some known relevance to colossal projection neuron fate specification. We're first on the list is a gene called SAPB2, which serves as a marker for colossal projection neurons. So if you knock it out, it doesn't show up in the end list. Another gene is called LHX2, which is a known upstream positive regulator of SAPB2. So we picked that up. This interaction has been shown before in mice. Another gene, TSHZ3, has been linked to cortical projection neuron development before and has ties to autism spectrum disorder, and a few other genes with functions mostly unknown that play some roles in the hematopoietic system, which we're following up on. And so with that, I told you about in vivo genome editing and functional genomics, how we can use Cas9 and Cas9 mice in order to perturb genes very efficiently in vivo. We can use these mice to deliver libraries and try to make genotype-phenotype connections, both in cancer as well as neocortical development. But now I want to transition to the next story, which is called transcriptional programming. And the motivation here is trying to understand cell state transitions. So this is a major topic of all of biology. What happens to my stem cell as it differentiates into a neuron? What happens to a healthy neuron as it transitions into a diseased neuron? So, and throughout this process, it's not just dictated by one gene, right? Previously, I told you we do screens where we knock out one gene and try to make connections between this transition, but it's obviously far more complex than this. It's this orchestra of transcription factors and other genes that dynamically change their profiles throughout this process. You know, some genes will go from low to high, others high to low, some transiently go off, or some transiently go on. And then we try to interrogate this process using single-cell RNA-seq, such as this plot shown here, which shows you the end states. But clearly, there's this whole trajectory that gets cells from point A to point B. And these single-cell RNA-seq experiments are purely based on association. These genes go up, but they don't get at causation. And this requires perturbation experiments. So we set out to develop tools that will help us try to recreate these profiles inside of cells, which is a very complex technology development problem. The question being, how can we write such a complex transcriptional program? How could I turn this or execute this program or force this program in a stem cell? It could be used for directed differentiation or causally validating in a single-cell RNA-seq experiment. Making these transcription profiles is quite complex. If you look at the genome editing toolbox, it's amazing. It's diverse. There's hundreds of papers. There's really good enzymes that can do just about anything really well. There's some CRISPR enzymes that can knock out a gene. There's some CRISPR enzymes that can activate genes or base edit. But what these tools can't do are unify these effectors and have a time element to them. There's no Swiss Army knife. There's no unified platform that can recreate these profiles. So what are the features that I think are important for recreating these profiles? I would say conditional. You need to be able to turn the system on. I would say inducible. You need to be able to tune how much you turn a gene on or off. I would also say orthogonal. It's not that you just want to turn a bunch of genes on or turn a bunch of genes off. You want to do some up, some down. And also, most importantly, I would say multiplexability. You don't just want to target one gene, you want to target several, and you want to do pathways. The solution to this gets quite technical and specific. So the way I'm going to show it to you is I'm going to show you the solution and walk you through some of the features. This work was done by postdoc Carlo Campa, as well as a PhD student, Niels Weisbach. And how we developed this unified framework is using an enzyme that is like Cas9, but it has a unique functionality. So the enzyme we use is Cas12a, which like Cas9 can cut DNA, being guided by a guide RNA, but it also has the ability to process its own guide RNA, its own cRNA, from a long CRISPR array, meaning you can supply this enzyme with a huge CRISPR array harboring a bunch of spacers. It has the ability to cut them out and use them for genome editing. So what we do is we deliver this construct inside of cells that basically uses a Pol2 promoter to express Cas12a. Then this is transcribed, and then eventually the Cas12a is translated. And what it does is the protein comes back to its own transcript, which we delivered, and is able to cut out its own guide RNA. And because we have this purple element called the triplex here, it stabilizes the upstream element, so the protein continues to be expressed. And does this work inside of cells, and how does it compare to the gold standard? This slide answers that. Where the gold standard is usually you deliver a CRISPR enzyme and then a guide RNA expressed from a U6 promoter. So if you do this inside of cells targeted to DNMT1 and HEX cells after transient transfection, you get about 25% editing rate, which is standard for Cas12a. But with our approach, if you deliver the guide RNA with a Pol2 promoter, we can achieve equal to or slightly better editing rates. Then if you combine that into the single transcript format, you can achieve approximately the same editing rates. So it shows that the technology can work. And why this is beneficial and necessary is that the EF1 alpha promoter allows us to use more complex expression mechanisms. We can use Cree, we can use inducible promoters, we can use the gauntlet of Pol2 promoters. And additionally, Pol2 promoters can drive really long transcripts, and the megabase are more range, meaning we're not limited on how many guide RNAs we can express. So now let me walk you through how we can use this single transcript platform, which we call SIT-Cas12a, and all these different features. So here's an example of where we express SIT-Cas12a under EF1 alpha LSL. So in the presence of Cree, we get editing. In the absence of Cree, we do not. So that's conditional. Then if we clone this construct downstream of a DOX-inducible promoter, either in the TET on or off configuration, we can tune how much editing we get it within a population of cells, so we can make it inducible. We can also make it orthogonal, and this is how we do that. So what we do is we use a catalytically active Cas12a, so it can cut, but we fuse it to a transcriptional activation domain, which has the ability to activate genes. And then we control whether it cuts and knocks out a gene or activates a gene based on controlling two components. One is the length of the spacer, either the standard 20 base pair format or a truncated 15 base pair format. And so if you take a full-length spacer and you target the enzyme to the exon of a gene, that enzyme will bind and cut, knock out the gene. But if you supply the enzyme with a truncated spacer, it will find its target and bind it, but it won't cut. And so if you target it upstream of a promoter, that transcriptional activation domain will drive transcription. And this is how, with the same effector protein, we can knock out or activate a gene, and that can achieve orthogonality. I'm just going to skip through this really quick. Turns out Cas12a transcriptional activators are quite poor, so we engineered a better version. And here are some data showing the orthogonality result, where if you look at the full-length CRISPR RNA, you can not activate a gene, but you can edit it. And in the truncated format, you get activation, but not editing. So it works as described. And then next, let's get at multiplexability. This is the desire to target many genes at once. In this experiment, we used a dead Cas12a fused to an activation domain. Supplied within this vector was 20 CR-RNAs targeted 10 genes. Here's a small RNA-seq showing that indeed each one of these CR-RNAs is expressed and processed. And then here's a qPCR result showing that each of these 10 genes that we targeted with these two different guide RNAs were significantly activated. And importantly, there was no pre-selection of the CR-RNAs. They were just designed using the standard software. And so it's quite robust for each gene. And so I just showed you we can make this CIT-Cas12a conditional, inducible, orthogonal, multiplexable. But what I haven't showed you yet is how we can combine them to get towards programming cell states. And so here's our final experiment I want to show you. And that is this experiment where we delivered the orthogonal construct expressing 25 cRNAs targeting 15 genes targeting 15 genes, 10 for activation, and 5 for knockout. So here are the 10 genes, two guide RNAs each that we can activate. And here are these 5 genes that we can knock out. So we can massively change the transcriptional and genome state within populations of cells. And this was done in a transient transfection experiment with these results shown 3 days after. So we're very excited by the promise of this platform. We can start to combine these unique features towards recreating these transcription factor or other gene dynamics with the long-term goal of being able to program cellular states and also validate some of these single-cell RNA-seq experiments. With that, I want to transition to the final bit, which is transcriptional recording. Now, this gets far away from neuro, but I think it's really cool, and it's going to have some potential in brain science, hopefully in the future. And this again gets at the question about transitions in cell states. How does a stem cell become a neuron? How does a healthy neuron become a diseased neuron? And how do we ask this question now? We perform time point experiments, right? At time 1, 2, 3, we break open different populations of cells. We perform RNA-seq, single-cell RNA-seq, and look at the genes that are expressed at different time points. The clear limitation here is that depending on when you look, you can see a very different picture. If you look early, you see the red genes on. If you look late, you see the blue genes on. And you have no idea what happens in between. You just have to perform inference. And so we set out to ask this question in a very technical way. How can we preserve this transcriptional information throughout time? How can we convert these curves into some memory format within cells that we can use sequencing to kind of reconstruct these profiles? How would we do that? If you remember from one of my very first slides, I told you about CRISPR-Cas adaptive immunity. And this first step is recording, where CRISPR-Cas enzymes are capable of physically excising nucleic acids, DNA or RNA, and inserting it within their own genome, where that serves as a molecular memory of that nucleic acid. And so a PhD student in my lab, Florian Schmidt, and I set out to try to turn this CRISPR-Cas recorder into a transcriptional recorder. It turns out it works, and this is how it works, where there's these two CRISPR-associated proteins, RT-Cas1 and then Cas2. RT stands for reverse transcriptase. Together they form this complex, and inside of cells, what we think is happening is they're randomly grabbing RNAs proportional to their abundance, converting them into DNA and inserting them within their own genome. Where what you see here is you get 3 different colored spacers, each one derived from a different RNA at a different time. giving us an entry point to reconstructing gene expression. And so if you turn on this transcriptional recorder in a large population of bacterial cells, isolate them and sequence them, and perform an RNA-seq-like bioinformatic analysis, this is what it looks like. Where what I'm showing you is the E. coli genome. It's 4.5 megabases. And on the y-axis, the number of spacers derived from that RNA. You see some peaks and some troughs. If we zoom in, we see that these spacers align to genes. It looks very much like an RNA-seq alignment. We can actually use this as an entry point to quantify how expressed that gene was throughout the duration of the recording experiment. And from a technology standpoint, it was very important to answer mechanistic questions. We know very little about how this RT-Cas1, Cas2 functions. There's a paucity of mechanistic details in bacteria. And so the first question that's really important is, are these spacers actually acquired from RNA, or is there just some other random thing going on? This could be answered quite easily simply by infecting some cells with an RNA virus and some cells not. And what you can see is that if the cells that are infected with this RNA virus, you get spacers or reads aligning to that virus, whereas in the control you don't. So indeed, spacers are derived directly from RNA. The next question that's clearly very important is that, is this information quantitative, or is it just a garbled mess? And so this is also very easy to answer, where we took our recording cells, so they had our recording machinery, and then we provided an inducible transgene. So this is just an inducible GFP. And if you induce it to different extents and compare the absolute mRNA copy number on the x-axis and our spacer count information on the y-axis, you get this really nice line with an R-squared of 0.97, showing that indeed that this information is quantitative in this context. And finally, and most interestingly, can we describe complex and transient events? Can we use this information to actually interrogate interesting biology? So what we did is we turned to a challenge experiment where we used this herbicide called paraquat, shown at the top, where it should induce a reactive oxygen stress response in these cells. So if you take different populations of cells and stimulate them with different concentrations of this herbicide and look at the transcriptional records derived from these E. coli, this is what the data looks like. Where here I'm showing you a Pearson correlation covariate matrix where each row or column is a sample. And what you can immediately see is that at the transcriptome scale, you have enough information to unbiasedly cluster them into these 4 groups. And they cluster according to the concentration of paraquat, meaning there's a lot of information in these records. We can also perform dimensionality reduction or principal component analysis, where each dot is a sample, a population of a large number of E. coli. Just looking at the top 500 most variable genes, we can cluster them unbiasedly into their three starting conditions, which again showing us that we can record this information. We can also perform a gene level analysis and ask the question, which genes are differentiating these samples using standard RNA-seq tools. We can build these signature gene heat maps, unbiasedly cluster them to their groups. We can also perform pathway analysis and kind of look at the genes orchestrating these responses. So here are some of these genes listed. Won't get into depth, but a lot of these genes that we find are fundamental to the behavior of E. coli to resist paraquat and ROS stress. And what I want to point out in particular is that you can see a dose-dependent increase in gene expression in these different samples, where here you have unexposed populations, here you have low, and here you have high. So you see this dose-dependent increase in gene expression on the transcriptome scale, which was very promising. This technique is scalable to other relevant physiological states within E. coli, such as oxidative stress and acid stress. We can unbiasedly cluster using at the whole transcriptome scale. We can use PCA, and we can identify all the genes orchestrating this process. And in the final set of experiments, what we wanted to do is try to see if we could capture transient events. So, imagine a gene that transiently comes on in response to an external stimulus. Can we capture this information in cases where an RNA-seq gets lost? So we set up this experiment where we had 3 populations of cells, some that were never exposed to the herbicide, some that were transiently exposed to the herbicide, and some that were chronically exposed to the herbicide. And we performed both our method, which we call record-seq, and we performed a standard RNA-seq protocol. And just for the sake of simplicity, I'm only going to show you the principal component analysis, and I want to draw your attention to what the transient paraquat samples look like. So with RNA-seq, the transient paraquat samples look exactly like the unexposed samples. So with RNA-seq, you have no idea that these cells encountered the herbicide. However, with record-seq, the transient paraquat samples cluster with the chronically exposed. So we captured that transient information. And if I had more time, I would provide you with a more in-depth analysis to show you that we could actually stratify in a dose-dependent way the transient samples and the chronic samples. So we get this cumulative sum information about exposures to the environment. With that, we created what we think is a very interesting tool in bacteria at this point that is synergistic and different to RNA-seq, where RNA-seq gives you these really high-resolution snapshots at individual times, giving you a wealth of information. But our technique allows us to look into the past by recording transcriptional information in the genomes of bacteria throughout time. With that, I would like to summarize and remind you this whirlwind of different vignettes that we talked about. I told you about in vivo genome editing and functional genomics, how we can use Cas9 and Cas9 animals to perturb genes in living animals and perform genetic screens, both in cancer and in brain development. How we can use this enzyme Cas12a to massively perturb cellular genomes and transcriptomes towards engineering transcriptional states. And also how we can hijack the CRISPR recorder in order to turn cells into transcriptional recorders at this stage, recorders of their environment, where our current goal is to try to use these cells as living diagnostics in people. And finally, the most important slide of the entire talk, the acknowledgements of all the incredible people in my lab and in other labs that have participated in this research. A huge thanks to Florian Schmidt, who was involved with both the in vivo functional genomics because of his AAV expertise, and also he drove the record-seq project in collaboration with Masha and Tenmei. Also want to point out Carlo Campa and Niels Weisbach, who led the transcriptional programming part, and everyone else in the lab who's just incredibly terrific. I would like to point out that the Cas9 mice and all the plasmid that we created are readily available from the Jackson Labs and Adgene. I'd like also to thank my incredible funding sources that allows us to do a lot of this wild research and create tools for the community. So thank you all so much for your time and attention. I would be happy to take any questions.
Thanks a lot, Randy, for this beautiful overview of your work. Quite inspiring in many ways indeed. I'd like to encourage people to ask questions either on the chat part of the interface or on the ask a question one, which is actually better because you guys can then rank them by perceived interest. Randy, what are the main challenges to molecular recorders in eukaryote cells as opposed to prokaryote ones? Is it a question of size? Is it a question of complexity of the genome, 3D structure?
We don't know. So us as well as others have tried to adopt some of these CRISPR adaptation systems into eukaryotic cells. We don't have a yes or no answer yet. It's complex. But it doesn't work the same as in bacteria. So we, as well as others, are currently trying to figure out this problem and seeing how we can make these CRISPR adaptation machinery work in eukaryotic cells. But there's no clear reason why they may or may not be working as well as in bacteria.
So what are the limits to the temporal resolutions of such systems?
What are the limits to the temporal resolution of the system? So currently, the efficiency of transcriptional recording, even in E. coli, is not terribly good. Recording only happens in 1 in every 1,000 cells, depending on the scenario. And we have to rely on very sensitive methods to extract this information. So what's limiting temporal resolution is the efficiency of these enzymes, which, due to how they evolved to not be very good. And if you can imagine a bacteria where the system was very good, that bacteria would be in a lot of danger. It'd be constantly just taking RNAs and shoving it in its own genome. The genome would constantly be expanding. And additionally, these are guide RNAs, which could be used to target oneself. So they're potentially autoimmune.
Mm-hmm.
So these enzymes evolved under very strict pressures. They can't be too good or else they'll just die, but they can't be so bad that they don't provide adaptive immunity. And so now our lab and others are working on protein engineering techniques to try to improve the system better than nature did.
Okay. One question from the audience: are there any strategies, e.g., continuous directed evolution systems, to improve capturing?
Yeah. So when an enzyme is bad, that's the first thing you want to try. You want to try to evolve it. There are a number of ways to do this. There are random, systematic, continuous. Just there's a whole bucket of methods, and we're trying several of them. I don't have too much to say at this point. It's ongoing stuff. We don't have anything, to say or distribute at this point.
Mm-hmm. What's your view on you mentioned state transitions? It's always a term that I have a hard time, picturing in real terms. At what point are you undergoing a state transition, or at what point do you call something discontinuous in transcriptional terms? Do you have any insights into that, or are you staying on the engineering part?
Well, I have no better insights than you or the rest of the community. Just cell state is a term that we use for lack of anything better. It's something that is very transcriptional module based. It's all that RNA seek. Can we differentiate cells based on their transcriptional profiles? Mm-hmm. Do they separate in a UMAP? Then they're in different states. What's the difference between a state and a type? Clearly, this is a very nuanced argument.
Could you easily envision systems that would be triggered by the combinatorial change in levels, relative levels of several genes? You say a 20% in that one, a 30% in that one, and a 40% in the third one. So dose-dependent systems with high granularity.
I mean, I can't say that I have much better insight than you on this one. It's a really complex problem. It's not just, it's really not just one gene. No, sorry, go ahead.
I meant in engineering terms, to have systems triggered by low level or average level combinatorial changes.
To try to tie genome editing to a particular cell state. Is that what you're saying?
Exactly. That's the better way to say it. Yeah.
So there would be ways to do that by expressing some of these enzymes and promoters that are linked to this state. If it's as simple as one promoter, it's quite easy. If it's a collection of promoters and relative levels, then it gets in the world of synthetic biology, and one could foresee a type of circuit that would only express a gene under certain conditions, and these conditions could depend on relative levels of mRNAs or microRNAs. So yes, I think this is engineerable using the tools developed in synthetic biology over the past decade.
So you could expect to be able to detect, say, 20% change in RNA expression levels to trigger something.
Yeah, so these type of synthetic biology gene circuits can be quite sensitive about levels in pure cell populations that don't fluctuate too much. If you're talking about, could you develop one circuit that would perform in all the diverse cell types of the brain, I think that's a bit of a stretch, but maybe within a certain class of homogeneous cells, one could imagine that it could work.
Okay. A question from Christopher Esk. Thank you, very nice. Can you control expression of the multiplexed activation knockout system, for example, in the temporal control of transcription factors in neocortex development?
I'm going to reread that. Yes, you could, using conditional or inducible systems. So imagine like a Cree-ER, a tamoxifen inducible system. So I showed you this slide where we can do a Cree inducible system. So you could turn on these, transcription factor profiles at a given time using something like Cree. You could also use an inducible promoter. If you aren't able to deliver tamoxifen, or if you aren't able to deliver docs or anything like this, you could also potentially express this Cas12a enzyme under the expression of a promoter that is turned on at a given time, right? It just depends on the specific area that you're talking about, but you could tie the cell manipulation to a particular state via expression of Pol2 promoter.
Okay. Maybe one last question from Shingo Nakazawa. Thank you for a wonderful talk. Is there any limitation or bias of transcriptional recording programming, for example, due to the accessibility of Cas to the target sequence locus?
So we spend a lot of time trying to understand the bias in the accessible sites in recording. The only reasonable biases we can find are that the sequences tend to be AT rich, which we think is because these RNA sequences tend to be less structured. And indeed, we see this genome wide, that we don't see a lot of heavily structured RNAs, which works heavily in our favor because most of the RNA in cells are these ribosomal RNAs and tRNAs. So we don't see a lot of these, which is really good. It means we record everything else, but this is a bias against structural RNAs. So AT rich against structure, and also we see areas in the transcripts that are preferentially recorded, which we think ties into the mechanism of this process. Where basically, if you have this long transcript, we mostly see spacers that are derived from the 5' and 3' end. And we think this has to do with how these Cas proteins interact with RNA molecules. We basically think they just grab the end, they protect the RNA from some cleavage event, and that serves as a footprint and dictates the size of the spacer, which we know is around 40 nucleotides in our system. Really great question.
All right.
Simona, thank you. You should ask a question.
Oh, yeah, there she is. I was going to say we're reaching the end. Simona, you have about 25 seconds to ask a question. All right. Randy, thanks a lot. It was awesome to have you here. We're reaching the end of the allotted time. Next week's speaker will be Fiona Francis from the IFM in Paris, and the host will be Stephanie Bolak from the ICM. Have a good week until then. Stay safe, take care, and see you around.
Thank you again. Thank you, everyone.